Welcome to this profound deep dive into the biological engineering of the human body. When we look at our skin, we often perceive it merely as an aesthetic covering, a barrier against the outside world. However, beneath the epidermis lies a vibrant metropolis, a complex system of molecular architecture that sustains our shape, our elasticity, and our youth. The great challenge we face, whether due to the passing years, sudden weight fluctuations, or gestational processes, is the compromise of this structural integrity. Stretch marks and sagging are not just marks. They are the visible result of a microscale collapse, a rupture in the foundations of the connective tissue. To understand how to reverse or soften these processes, we must stop seeking superficial solutions and begin to understand the science of tissue regeneration from an integrative perspective. To maintain skin firmness, we depend on a specific cellular lineage that works tirelessly in our dermis, the fibroblasts. Imagine them as the engineers and laborers of a major construction site. These cells are responsible for synthesizing the extracellular matrix, a complex tangle of proteins where collagen and elastin stand out. Collagen acts like the steel beams of a building, providing tensile strength and preventing the skin from tearing under pressure. Elastin, on the other hand, functions like springs, allowing the tissue to stretch and return to its original position without deformation. The problem arises when the demand for stretching exceeds the synthesis capacity or the quality of these proteins. When a fibroblast is malnourished or lacks correct signaling, it produces fragile collagen, leading to what we know as the snapping of the fibers, the genesis of the stretch mark. This is where the silent protagonist of our biology enters, organic silicon. In the chemistry of life, silicon is not just a trace mineral, it is the cross-linking agent. Without silicon, collagen and elastin fibers are like loose threads that cannot weave together to form a strong rope. It acts by stabilizing the macromolecules of the extracellular matrix and functions as an essential cofactor for hydroxylation enzymes, which are responsible for giving the collagen molecule its final shape. At the molecular level, silicon ensures that the skin structure is organized and resilient. When silicon levels drop, which occurs naturally after age 25 or 30, the skin loses its capacity for efficient self-repair, becoming more susceptible to permanent ruptures and loss of density. When the rupture that generates the stretch mark occurs, we enter a complex pathophysiological process. Initially, we have the inflammatory phase, red or purplish marks, where the body is still trying to send blood flow and defense cells to that internal wound. Over time, this inflammation subsides and gives way to white marks, striae alba, which are essentially scar tissue, a frustrated attempt by the body to close the gap with low-quality, dense collagen that lacks elasticity. The transformation we seek is to reactivate cellular signaling in these areas. By providing the correct supply of silicon, we are essentially restarting the fibroblast machinery, allowing them to replace this disorganized scar tissue with younger, more functional fibers, significantly improving the texture and coloration of the affected region. One of the most effective and ancient ways to promote this mineral delivery is through biophysical intervention with white clay, scientifically known as kaolinite. White clay is notable for its high concentration of silicon dioxide and for having a pH very close to human skin, which minimizes irritation and maximizes biological acceptance. When we apply a clay poultice, a phenomenon of ion exchange and adsorption occurs. The occlusion created by the clay paste increases local temperature and hydration of the stratum corneum, opening pathways for trace elements, especially silicon, to penetrate through a concentration gradient. This is not a cosmetic miracle, but a mineral diffusion process where the clay serves as a substrate reservoir that the skin absorbs to feed its internal regenerative processes. Concluding that skin health depends only on expensive creams is to ignore the beauty of our own biology. True firmness and tissue regeneration are the results of conscious biological engineering. By uniting the understanding of the role of fibroblasts, the chemical importance of silicon, and the practicality of natural resources like white clay, we move out of the field of trial and error and into the field of evidence-based integrative health. Remember that the human body possesses an intrinsic capacity for healing, provided it receives the correct building blocks and the necessary time for tissue remodeling. Caring for the skin under this lens is, above all, an act of respect for the mineral and cellular complexity that keeps us whole and resilient in the face of time.